Hello, big fella. Today we will study about history and development of microbiology. But, what is microbiology?
Microbiology is the study of microscopic organisms, or microbes. Microbes are found everywhere in the environment and play a vital role in our planet. They can be beneficial, such as helping to produce food and medicine, or harmful, such as causing disease. Microbiology is a rapidly growing field with applications in a wide range of industries, including healthcare, agriculture, and environmental science. The development of microbiology as a discipline can be traced back to the invention of the microscope in the early 17th century. Antony van Leeuwenhoek, a Dutch draper and amateur scientist, was one of the first people to use a microscope to observe microorganisms. He made detailed drawings of bacteria, protozoa, and other microbes, which he called animalcules. Leeuwenhoek's observations sparked a great deal of interest in microorganisms, and scientists began to study them more closely. In the 18th century, Lazzaro Spallanzani and John Needham conducted experiments that showed that microorganisms could not arise spontaneously from non-living matter. This disproved the theory of spontaneous generation, which had been widely held for centuries. In the 19th century, Louis Pasteur and Robert Koch made significant contributions to the field of microbiology. Pasteur showed that microorganisms could cause fermentation and spoilage, and he developed methods for preventing these processes. Koch developed a set of 4 postulates that are still used today to determine whether a specific microorganism is the cause of a particular disease. The work of Pasteur, Koch, and others led to the development of the germ theory of disease, which states that diseases are caused by microorganisms. This theory revolutionized the field of medicine and led to the development of new treatments for infectious diseases. In the 20th century, microbiology continued to grow and develop. New techniques were developed for studying microorganisms, and new microorganisms were discovered. Microbiology became an important discipline in many fields, including healthcare, agriculture, and environmental science. Here are some of the major milestones in the development of microbiology as a discipline. 1674. Antony van Leeuwenhoek first observes microorganisms using a microscope. 1765. Lazzaro Spallanzani disproves the theory of spontaneous generation. 1857. Louis Pasteur discovers that microorganisms cause fermentation. 1876. Robert Koch develops the 4 postulates of germ theory. 1881. Louis Pasteur develops a vaccine for rabies. 1928. Alexander Fleming discovers penicillin. 1944. Oswald Avery, Colin MacLeod, and Maudslin McCarty discover that DNA is the genetic material. 1953. James Watson and Francis Crick determine the structure of DNA. 1977. Stanley Cohen and Herbert Boyer develop recombinant DNA technology. 2003. The Human Genome Project is completed. These are just a few of the many important milestones in the development of microbiology as a discipline. Microbiology is a rapidly growing field with a bright future, and it is sure to continue to make significant contributions to our understanding of the world around us. Now let's talk about 2 theories: spontaneous generation and biogenesis. Spontaneous generation is the theory that living organisms can arise from non-living matter. This theory was widely held for centuries, and there were many examples that seemed to support it. For example, it was observed that maggots would appear on rotting meat, even if the meat was kept in a sealed container. It was believed that the maggots had spontaneously generated from the meat. Biogenesis is the theory that all living organisms come from pre-existing living organisms. This theory was first proposed by Francesco Redi in the 17th century. Redi conducted a series of experiments that showed that maggots did not spontaneously generate from rotting meat, but instead hatched from eggs laid by flies. In the 19th century, Louis Pasteur conducted a series of experiments that further disproved the theory of spontaneous generation. Pasteur boiled meat broth in a flask, and then sealed the flask with a swan-necked neck. The broth remained sterile, even though it was kept in a warm environment where microorganisms could grow. This showed that microorganisms could not arise spontaneously from non-living matter, but instead had to come from other microorganisms. The work of Pasteur and others definitively disproved the theory of spontaneous generation and established the theory of biogenesis as the correct explanation for the origin of life. Today, we know that all living organisms come from pre-existing living organisms. This is a fundamental principle of biology, and it has important implications for our understanding of the natural world.